Okay, well, shall we get started? Okay, well, I just want to welcome everybody to the United Leukodystrophy Foundation's virtual family conference session on CTX, presented by Dr. DeBarber, Dr. Vaz, Dr. Larson, Dr. Eichler, Dr. Bonin, and Dr. Gelb. This session is being recorded and will be posted for viewing online in early July. Thank you so much for joining us. And now I will turn it over to Dr. DeBarber. Thank you.
Okay, so welcome everyone to this session today. Before I introduce the speakers, I would like to dedicate this session to our friend Marc Chevrier, who very sadly passed away earlier this year. So in addition to being a lupus research hero and a pioneer and patient advocate in the field of lupus, Marc became a CTX patient advocate after his family was touched by CTX. I first communicated with him in 2014 when he reached out to see how he could assist in CTX research efforts. We then met in person for the first time in 2016 when he attended the ULF annual meeting, as well as the first meeting that was held to discuss nominating CTX for consideration for addition to the RUSP. And that was where Marc presented a case series describing his family story. So Marc was a devoted family man, incredibly kind and generous, and such a wonderful person to have as part of the ULF and CTX community. He was incredibly smart but humble, with still so much to contribute and gone far too soon. Me personally, I will really miss him and the conversations that we used to have at the annual ULF meetings. And I know the CTX community as a whole will really miss him as well. So Marc, the meeting today is dedicated to you. And I am thrilled today with the diverse and interesting set of speakers that we have. And thank you so much, everyone, for agreeing to give a talk for this CTX family session. And especially Fred, who I know it's 11 p.m. in the Netherlands, so we really appreciate it. So speaking today, we have Bobby Blanchard, who is a board member of the CTX Alliance, a CTX patient advocacy group. We should have Dr. Eichler joining us from Mass General, Harvard.
He's here.
Fantastic, to talk about formation of the CTX Medical and Scientific Advisory Board. Hopefully also Dr. Larson, who will update everyone on a systematic evidence that he and a group of other scientists is working on CTX diagnosis and disease modifying therapies. Then I will give an update on the resubmission of the CTX nomination for consideration for addition to the RUSP. And then we have Dr. Vaz from the AMC Amsterdam, who'll give an update on the Netherlands pilot study screening newborn dried blood spots for CTX. And then Dr. Gelb from UW will talk about the Screen Plus pilot study screening New York State newborn dried blood spots for CTX. And then finally, we'll have Dr. Bonin from Bile College of Medicine, who is going to give us an update on the prevalence of CTX and genotype, phenotype information for CTX. And unfortunately, there will not be, the session will finish at the top of the hour. So we'll have to finish promptly. So I would ask the speakers if we can try and stay on time so that we can leave a short amount of time for questions at the end. That would be fantastic. So with that, I will pass over to Bobby Blanchard.
All right. Well, hi. Hello there. My name is Bobby Blanchard, and I have the privilege of announcing the new organization for CTX called CTX Alliance. there's a nice little photo there on all of us that have been coming together the last several months and trying to iron out some of the beginnings here. It's kind of exciting. So, and I look like I have the, oh, here. And just briefly, I do have a short little video. I hope it comes across.
I'm Tim.
I'm Jace. This is Trini. I'm Joel.
I'm Jordan.
I'm Bobby. And together,
We are the Planters.
Oh, yeah. Don't turn on the camera.
All right, so those are my people. If you missed it, it kind of went fast. We have 2 children affected by CTX. And we were just really blessed to have restarted treatment and got a diagnosis fairly early on. And our children are doing wonderfully. So we kind of feel a strong responsibility to just try and find those that are undiagnosed. And we know that we have a great treatment available. Of course, we want to cure, but we also want to support those who maybe aren't in the same situation that we are blessed to be in here. So that's why I'm involved. But when we all came together, talking about a purpose for the CTX Alliance, we came up with our purpose statement of to provide education, support, advocacy, and promote research for patients affected with CTX, their families, and medical professionals who treat and study this rare disease. I think we noticed a lot of common things. We have been wonderfully supported by the ULF and Hunter's Hope and some of these umbrella organizations. But we know that CTX is slightly different than a lot of the leukodystrophies. And we saw that there were just opportunities that we could just maybe do better in the future for reaching people and just a way for families to find information more quickly and easier than they have in the past. And there's our mission: to support, educate, and provide a voice for CTX patients and their caregivers as we work toward a cure. As I mentioned, we just started in the last several months to work on this. And so, and it is a long process for this 501c3 that we're kind of finding out. But we have had a tremendous amount of assistance and help, and we're really grateful. But we have so far filed the articles of incorporation with the state of Illinois. We have drafted organizational bylaws. We are in the process of recruiting board members. We're filing for that 501c3 status with the IRS and other licenses that we need. And just thinking about future development of activities and programs and kind of these key areas: research, information, and support. And we just want to, things like newborn screening and that has come such a long way, but just seems to take such a long time. And then making sure we have information for patients. I know as a CTX family, finding information was incredibly difficult for us in the beginning. And within like our CTX community, like on our Facebook group, that was consistently one of the difficulties that people had, was finding physicians, the right physicians. What kind of physicians do people see? How often do they have certain things checked? And there is a lot of discrepancy kind of across the board. And so just trying to make some of those things easier, especially for newly diagnosed patients and their families, I think would be just a huge help and a huge gift for the CTX community. So that is kind of the update and where we're at with the CTX Alliance. One of those people who has helped us so much in this process is Jean Pickford, who is also on the call today. So if there's anybody who would like more information or would like to get involved, I mean, we are literally just starting, so it's a great time to jump in and get involved and help, yeah, get this started. There's her information there.
That's fantastic. Well, thanks very much, Bobby. That's great. And so now I would like to introduce Eichler, who's going to talk about formation of the CTX Medical and Scientific Advisory Board.
It's a pleasure to be here, and nice to see Andrea and Bobby and follow them. I feel it's a really terrific time for CTX, and seeing that there's a new patient community coming together, and also that we are recognizing that bringing specialists in the field of CTX together can add really much to this field. And I think the synergy between this academic effort and what Bobby just talked about could be terrific. And so I wanted to just briefly introduce something that is really just in the making. And Andrea has kindly helped organize and chaperone some of the calls we've had, and I will present how I think about this and what potential is available here. So this is a picture of all of us at a recent call of this Medical Scientific Advisory Board Executive Committee. We bring together really a broad array of experts.
We don't see the slide change, Florian.
Oh, you didn't see the slide change?
Yeah. You'll see the first slide.
You still see it. Sorry. I'll stop share and start again.
Yeah, it's okay now.
Now it's good. Okay. Now you can see all our pictures here.
Yeah.
So the idea here is really that a disease like CTX requires a broad array of different perspectives, scientists, physicians from different disciplines, because the disease affects different domains. It affects not just the biochemistry, but also the nervous system, and there are problems with gait and balance that need to be addressed by a physiatrist. There are GI issues that need to be addressed by a gastroenterologist. There are questions around cataracts that need to be addressed by an ophthalmologist. And I think over the past year, we've recognized that if we ever want to get to something like a cure, we really need to tackle this from multiple angles and bring all stakeholders together here. And so we were very fortunate to have really experts in the field who understand the biochemistry, like Andrea DeBarber, Bill Rizzo, Robert Steiner. We have some new members here of our Medical Scientific Advisory Board, Austin Larson, and then experts like Brian Wishart, who are bringing an understanding of the physiatry and the daily needs around ambulation to this. I think for many years in the field of CTX, we've had a great biochemical insights and a really noble effort around reducing the biochemical culprit of cholestanol. And that should continue. But at the same time, we have to understand that despite this biochemistry being consistently there are different stages of disease and different parts of the body that are affected different ways. And trying to bring everybody together here who can help address this will serve sort of the broader needs of patients beyond a focus on the biochemistry alone. So the mission statement here of the Medical and Scientific Advisory Board is to provide an independent and accessible academic home for the CTX community with the goal of improving the lives of CTX patients and their families, promoting early diagnosis, and raising awareness of the disease and its unmet clinical needs, supporting CTX research, and bringing together all stakeholders with an interest in curing this disease. So here, I think some of the key words is we want to be independent from industry efforts, but we want to collaborate with industry efforts. So I think that's an important distinction. It doesn't mean that you don't work together, but it means that you are an independent voice. And I really like that about Bobby's presentation, that she spoke to the voice of the patients. And I think that's a key ingredient that is needed here. And likewise, we want to be the scientists and physicians who help that voice be heard and add the medical knowledge and facilitate the kinds of treatments and trials that are necessary, recognizing that beyond the ultimate cure, whether it's a genetic or biochemical correction, there are day-to-day needs that patients have that need to be addressed by a broad community. It starts with the beginning of life and the early diagnosis and newborn screening that Andrea has been working tirelessly for, but it goes all the way through understanding early development, understanding academic cognitive needs of children growing up, what are the early signs, what are the early discomfort, how is diarrhea disrupting daily life, how are tremors, how is poor balance not making a patient be able to fully participate in the community, and how are things like xanthomas and their surgery impacting a patient's lives? What kind of guidance can we provide for management of those xanthomas? And instead of each patient trying to figure that out on their own, we want to be a forum where families can come, ask questions, and we look at the evidence and we provide guidance and we grow together. Ultimately, of course, we have the interest in fixing this disease, but knowing that fixing this disease completely will require a group effort and ultimately means different things at different stages of life and different stages of the disease. And we don't want to leave anyone out here just because you might be too advanced for a trial or you're wheelchair bound doesn't mean that you don't have needs that should be addressed. And so this is a part of, I think, our effort to bring all stakeholders together, create an inventory of all symptoms, all needs, and align that unmet need with the biology around this single gene. So I mentioned earlier that the various members to this Medical and Scientific Advisory Board I want to mention that we're an open community that is trying to bring together all the stakeholders that are needed. We're looking for people who are interested in CTX, physicians who have expertise in ophthalmology and surgery and other aspects, who want to advise us, who want to be a resource for the CTX Alliance. We're thinking about different ways to go after what we perceive as low-hanging fruit. Short-term goals may be to develop medical knowledge base and educational materials, webinars, and I think this is something where we could work closely with the patient and patient advocacy group, because hearing their voice and understanding their needs is going to make this medical knowledge much more accessible. We clearly have to do this with the audience in mind and understanding, whether what the medical information we're providing is addressing the key questions and priorities patients have. I think over time, what we would like to see is a real network of physicians arise that provides access for patients in the right geographic distribution. Right now, a lot of patients feel left alone, isolated. They have to travel long distances to find a physician expert. Are there ways that we can create alliances also with a GLIA CTN network that I am part of, that is a multi-center clinical network where experts in leukodystrophies are providing care and standardizing assessments across centers. As we're learning things for other adult onset neurologic diseases, this might be applicable to CTX as well. Are there patient resources that are coming about for other diseases that have trials or good symptomatic treatment that we can also bring to the field of CTX? So I'm hoping this is, Bobby, really a standing invitation for you to participate here and help and guide also the medical advisory board as to the needs and priorities that patients have. And likewise, we hope that you will also look to us to address some of the medical needs that you see. In order to get to trials, often what is needed is a robust natural history. And these are kinds of natural history studies that we're rolling out now in the GLIA CTN, which is a clinical trial network for leukodystrophies. And clearly having newborn screening up and running in the future will allow that to happen from birth across a lifespan. But even starting now with knowledge and inventory of patients who are living with CTX and what their daily disease burden represents is key. So just to mention contact information, you already heard of Andrea DeBarber at Oregon Health and Sciences, myself at Mass General, together with Brian Wishart, a physiatrist who works closely with me, Bill Rizzo at Nebraska Medical Center, Austin Larson, Children's Hospital in Colorado, and Robert Steiner at University of Wisconsin. So this was just a brief introduction, and Andrea, please add to what I said and correct anything that I left out.
That's fantastic. Fantastic. Thanks very much, Florian. We are going to move along and leave questions to the end. So I will move promptly into, oh, actually, okay, Dr. Larson is here. Okay, fantastic. So I would like to introduce Dr. Austin Larson, who is going to talk about a systematic evidence review that he and a group of others have been working on.
Hi there. So I'm Austin Larson. As you can probably tell, I'm sitting in a clinic room at Children's Hospital, Colorado. So up until about 1 minute before this meeting, I was seeing a patient for a newborn screen result. So I'm on call for the metabolic genetics group here. And I think that really puts me in the mindset of what do I need to know when I'm seeing a patient who has an abnormal newborn screen result. And so that's kind of the driving thought process behind this systematic evidence review, is to create the resource that will provide the evidence that's needed to know how to appropriately proceed when you get an abnormal newborn screen result. Hopefully, in a few years, the children in many or most or all states in the country will be tested for CTX as part of the newborn screen. And what do we need to know as the next step? So certainly, having done newborn screening, not myself, but as a community, having done newborn screening for the last 15 years for a variety of metabolic diseases, you realize that when you scale up the number of people that you're testing for a diagnosis by 3 or 4 or 5 orders of magnitude, you find a lot of gray area and a lot of nuance, and you really need a rigorously developed evidence base in order to know how to proceed when you're in those gray areas. So over the last year, we've identified the questions that are probably going to come up when we identify patients with CTX on newborn screen. And I should add, this will be appropriate for patients that are diagnosed by other methods as well, not on newborn screen. But the questions that will come up and the things that clinicians and other providers need to know in order to know how best to take care of their patients with CTX are the basis of this systematic evidence review. And so over the last year, we have developed a methodology for how we're going to review the literature. We've assembled a team of content experts. We have experts in the methodology of systematic evidence reviews. And as of recently, and with the support of Hunter's Hope, we've been able to hire a couple of researchers to do the hard work of digging into the hundreds of papers that have been published about CTX and to pull out the relevant details from each individual patient and assemble that into a database that we can then query and use that database as the basis for disseminating the state of the evidence about CTX. And so the specific topics that we want to cover are how do you accurately diagnose CTX? It's pretty clear at one end of the spectrum, but we know that with all metabolic diseases, there's a gray area, as I said. So we want to really solidify what it means to have a diagnosis of CTX. We also want to know what the basis for therapy should be. So how strong is the evidence for pre-symptomatic therapy? Should the therapy be driven by the relative elevation of particular biomarkers? Should the therapy be driven by different genotypes? Those are all questions that we're going to try to get at. We also want to compile the evidence for just how effective is the therapy. What are the aspects of disease that are most impacted by starting therapy? How should that therapy be driven? Should there be a biochemical target for therapy? Are there clinical endpoints for therapy? Does pre-symptomatic treatment truly prevent all symptoms? And over what time course? Is that true for years? Is it true for decades? We want to compile all the evidence to determine that. And lastly, if we are choosing to start an infant who doesn't have any symptoms, if we're choosing to start them on a therapy, there's a different risk-benefit calculus that we have to consider. So we have to very carefully consider the safety of currently available therapies. So I'm happy to talk more about those topics in the Q&A at the end, but I will turn it back over to Andrea for emceeing.
Thanks very much, Austin. That was great. Okay. to keep us on track, I will go straight into my presentation. Today I'm going to give an update on the nomination of CTX for consideration for addition to the RUSP. My disclosures are honoraria from Leading Biosciences and Travere Therapeutics and grant funding and so forth. So I just briefly kind of like to remind everyone of the timeline of the nomination. We were just discussing before the meeting started that in July 2016 was the first meeting that was hosted by the ULF to discuss submitting the CTX nomination for consideration for addition to the RUSP. And then with some methodological advances, you know, in 2017, Fred demonstrated that newborn dried blood spots can be identified using the peak area ratio between tetraglucuronide species and Toro CDCA that he'll be talking about today. And then in August 2018, the nomination was submitted to the Secretary's Advisory Committee. And then about the same time in 2018, my lab confirmed the primary tetraglucuronide species structure and identity. So we were able to synthesize authentic standard and stable isotope labeled internal standard for performing more quantitative analysis. And then in November 2018, there was an in-person Secretary's Advisory Committee meeting in Rockville, Maryland, where the CTX nomination was discussed. And the nomination was tabled until additional evidence could be provided. And here is members of the nomination team that travelled to Rockville, Maryland to attend the committee meeting and provide public comment. And so the advisory committee responded and laid out a plan for evidence that they require before the nomination could be resubmitted. And they agreed that CTX was a medically serious nominated condition with approved confirmatory diagnostic process and defined treatment protocols. So that was good. In terms of some of the evidence they were looking for, you know, some of the most critical evidence is that they're looking for prospective pilot data from population-based assessments. So they want the prospective screening of newborns to see if the methods that we have to screen newborn dried blood spots would work and they could identify a identifiable CTX newborn. And Dr. Gelb can touch on this as well later in the session. Also, they would like to see more analytical validity data for the screening tests and characteristics of the screening tests, that they're reasonable for newborn screening. They wanted to see more evidence on the case definition and the spectrum of the conditions to help predict the phenotypic range of children who will be identified based on population-based screening. And do the results have clinical utility? Do the genetic results or the biomarker results from newborn screening, can they help the physician know whether, for example, to treat the infant positive identified infant? So to go into each of these items in more detail, the first one being the case definition and the spectrum of the conditions. They asked if these are well described in CTX to help predict the phenotypic range of those children who will be identified based on population-based screening. And so at that point, the evidence, there wasn't enough evidence to determine this. And also in terms of in the response that they provided, they talk about a case definition that we need to add clarification regarding what is considered a true positive CTX case and include relevant biomarkers if applicable and references or documentation of consensus amongst experts in the field. So we've been working to try and make this evidence available, and you can see a few very important papers that have been published in the last year. So this group here published on a CTX case in a family of two siblings where they don't have the biochemical findings that we've normally seen in the past. So some of the other markers that are associated with CTX were elevated in these individuals, but not cholesterol. So this is, you know, very important to get to this spectrum of CTX, and are there potentially milder cases of CTX? also. And then an important paper to come out of Adveripsis group is that they describe a cohort of patients, and these are different patients from different families and around the world with different genetic backgrounds and no link with the biochemical markers. But these are cases of CTX in adults without neurological involvement. And this is really important because CTX has always been considered to be a progressive neurological degenerative disease. And so this is the type of information that the committee will evaluate for newborn screening. And then there are a number of different groups working on systematic evidence reviews, and Dr. Larson just gave a talk on one of them, to help inform the newborn screening process. And Dr. Bonnen will be talking today about some of her work to, help provide information to answer these questions from the committee. And then also, do the biochemical or genetic results that would be attained from newborn screening, could these help inform treatment? As Dr. Larson said, how does a physician know whether treatment should be started in a newborn that doesn't yet have any clinical symptoms of CTX? Another thing that the evidence that the committee was looking for is how early treatment can impact health outcomes. And so this is very difficult evidence to generate without newborn screening already. So I'm working with Dr. Philippe Zakai in Israel, where they have been performing prenatal screening for many years in the Druze population that has a higher prevalence of CTX. And I'm hoping that she will publish her data on this. I did present a poster with her at the ASHG meeting in 2018 on her experience with identification of CTX in newborns in northern Israel. Newborn screening followed by treatment from birth onwards provides a functional cure. And so that was 10 years of follow-up after identification. So hopefully we can get that data published as well. And then in terms of the test characteristics, analytic validity and of the tests. And when we submitted the nomination, there was Fred Vaz's initial pilot study in the Netherlands. My group had performed a small pilot study in Druze newborns, and Mike Gelb was working on a larger pilot study screening anonymous Washington State newborns. And so in the interim, all of these have now been published, including Mike's study. And so this data will be available for the committee. And then, one of the most critical pieces of data is their prospective pilot data available. And so I'm very glad Fred and Mike will be able to give us an update on this today. And so there'll be, as I mentioned, Mike's paper was published in June 2020 on his pilot study screening more than 32,000 Washington newborns. And Fred will talk today about his pilot study in the Netherlands. And so in terms of resubmitting the nomination, we're really dependent then on a newborn being identified in one of these pilot studies. So it's hard to say when we will be resubmitting the nomination, but it will be after, it's dependent on identifying a newborn in this process. And this is the original team that put forward the nomination of CTX, this group of stakeholders. And so we will start to meet up again. I'm hoping to have 2 to 3 meetings per year so that we can try and keep the momentum going. And, I'm hoping more people will be willing to join this team. So if you're interested in supporting this effort, please drop me an email so you can be involved in supporting these efforts. Okay, and with that, I will hand straight over to Fred.
Okay. Okay, I hope you can see my screen now. Okay, the Dutch experience. Is CTX newborn screening on the way? Well, I hope so, and I do think so. Let me see if I can advance this. Yes, so why are we here? The short version is, in the Dutch screening program, the official Ministry of Health considers CTX a serious candidate for newborn screening. This was in 2015, but there was no test. So we developed it in 2016. We partnered with Mike Gelb and Andrea de Barber to get CTX into the RUSP, or at least to support this effort. And well, Mike will tell you about this. He measured a lot of blood spots with excellent results in 2020. We, the Dutch, are now running a second pilot, 20,000 blood spots. And I'm here to tell you how we're doing. Of course, I cannot, sorry, Florian, I have some, cannot have it without any biochemistry. We'll do that very quickly. Show you briefly how the test works, what we are doing with these 20,000 blood spots, brief first results, sneak peek, and my hope on what is to come. So the plasma bile acid profile of a no CTX on the left and that of a CTX patient on the right. You see there's a very clear difference. So that's what I thought a long time ago when we diagnosed a very young infant by chance with CTX with this profile actually, which was treated immediately and is now doing very well. And this prompted us to think, should we develop an assay with this biomarker in mind? And these are bioalcohols. So where do bioalcohols come from? Let me see if I can get a pointer. That's nice. Okay, I want to do a complete biochemistry lesson, but briefly you see here cholesterol, and the all means it has one hydroxyl group. And actually what you do to convert it into the bile acids, quinodeoxycholic acid and cholic acid, you add a lot of the hydroxy group and a acid. So 1-ol, diol, triol, and then you hit a bump, and this is the CTX blockage, which holds the metabolism at the trial. So when this trial accumulates, it cannot go further, and the body frantically starts to add more of these hydroxyl groups to get rid of these metabolites that are accumulating and are potentially toxic, and so that you can excrete them in the urine, so pee them out. And you can even get 5, 6, 7, so pentols, hexols, and so on. So the most abundant one is this one. This is the tetrol. So this is one of the 4 hydroxyl groups. And actually what you see here is a glucuronic acid conjugate, meaning that the body adds another sugar-like molecule, which is very water soluble, which makes it even easier to pee out. And you don't need any derivatization to measure this, so no chemical modification before the analysis, which is very good. But unfortunately, measuring tetrol alone is not specific enough, or well, we might come back to that, but at least it wasn't at that time. So the trick, what we thought of for CTX was that, well, because this tetrol is accumulating, and we know that these bile acids are low, the end products, which you cannot make, because, well, we see this also in CTX. So there's a lot of pilots that we ran. You see in CTX, the CDCA level is very low. We know this. And that if you divide tetrol, the accumulating one, by a CDCA, the, well, very low one, you get a exponentially high or a very high ratio. And it's indeed the case. So these are the same data. And you see where no CTX, this ratio, the tetrol divided by the taurine conjugate of CDCA is highly elevated. So this is a good biomarker. So what we did is we decided and got a grant for that to measure 20,000 blood spots. And we did it in 2 ways. So we directly infuse it in the mass spectrometer after we extract the metabolites, and we do it via an LCMS. So first we separate it on a column and then do the MS analysis. So we now are at 16,000. And we didn't find a CTX patient, which is not very surprisable because it's about 1 in 100,000. We'll hear about that later. But let me show you some results. So briefly, only the LCMS. So this is the tetrol concentration of 16,000 individuals, newborn cards, anonymous. And you see the tetrol, okay, nice. And there's no one that is higher than 500 nanomolars, the cutoff that we used with Mike in the paper. So here, show me the controls we used. So we have 2 CTX patients with well, variable levels of the tetrol, and you can already see that on basis of the tetrol alone, you can, well, quite separate them out. There was only, so as I said, 1 tetrol over 300 nanomolars. And what we did is we looked at the ratio, which you will show in the next picture, and concluded that this wasn't a CTX patient. For the ones that are here, we said, okay, let's check them, and they all have this ratio that is very low, meaning that there's no CTX. So let's look at the other marker, the one that we thought about. So here's again the plot of all the tetral ratio, CDCA ratios. And if you put them again at the side with the CTX control, you see that there's complete saturation. Again, we didn't find any one that was higher than 0.3. If you look at all the top ratios, all have normal tetral values, but and low CDCA values, meaning that the ratio is a little bit higher, but not as high as you would say. We still have to determine the cutoff, but at least they're not CTX based. So, summary, assay looks promising. We have no positives in 60,000 blood spots, suggests that's a good method for newborn screening. Both direct infusion MS and LCMS option are possible. But for the direct infusion, I only show you the best results, but the other ones are pretty good too, but you need the additional ratios that we described in our paper to really get a clear separation without any positives. So what's next? I'm almost done. Finish the 20,000, obviously. Then we formally submit the CTX test as a suitable newborn screening test in the Netherlands. And the RIVM already has said that if this is a success, that they will try and get it to a national trial. So really start screening. We don't have a lot of people in the Netherlands, only about 70 million. So we have about 140,000 births a year. But still, if we get to that stage, we will have a prospective screening pilot, not even a pilot, it will be an actual screening, then we actually will have started. We're not there yet, but that's what I'm really hoping for. Of course, we will continue to work with all of you, Andrea and Mike especially, to get CTX into the RUSP. And then my last slide is to show you all the people that worked together to get all this stuff done during the years and who financed it. So was that fast enough?
Yeah, that was perfect, Fred. That was I'm so excited by the data. This is really fantastic, and I'm so glad that everything's going so smoothly and that it's looking like it works so well so far. So I do have questions for you, but I will save them up to the end. And now I'll hand over to Mike.
Can you see my slides and hear me?
Yep.
Okay, thanks, Fred. Yeah, I'm gonna jump to CTX here. Yeah. So basically what happened is we were running a large pilot study in Washington State on MLD, funded by Takeda and NIH. And it's LC mass spec, about 2 minutes per baby. And we were measuring sulfatides and dried blood spots. And that was published, it works great. Andrea calls me up and says, Mike, we're working on CTX. We'd like to pick your brain. And I said, well, look, we're running this. So we'll just open up a detection channel for these CTX markers and just pop it into our MLD pilot basically for free. It was trivial. We did that. So we started doing that. And we ended up deciding that the Dutch tetral marker that you just heard about was quieter. We were getting essentially zero amounts of it in thousands of newborn blood spots. And the marker that Andrea was working on, another lipid marker, was a little bit noisier. So we decided to go forward with the Dutch marker first here and to use the de Barber marker sort of second here. So to make a long story short, it works like a charm. So we did about 50,000 de-identified blood spots. So why de-identified? Because you need parent consent if you're going to identify the babies and tell them, maybe that you're screened for this disease that wasn't on the panel, and now your baby has CTX, and maybe they don't like that. So you need the consent, and we have no way to go around the country, go around Washington State and consent all these birthing centers. Okay, so we just get 1 month old dried blood spots from Washington State as babies are born, and we have state IRB approval to do a research study as long as we keep the identification of the babies secret. So we did 50,000, and we set a cutoff of the VOS marker based on about 7 dried blood spots that Andrea got from patients with CTX. And these have to be newborn blood spots because we care only about the level of the marker in newborn blood spots, right? Not already diagnosed older patients. And the marker is incredibly quiet. We only had 1 newborn above the cutoff. And you see in Fred's talk at 16,000, he has essentially none. So no hits. We had 1. So it's a really quiet, good marker. We did DNA second tier on the same blood spot, and it confirmed it was CTX. It was a very clear genotype. This genotype has been seen in the clinic. That means this combination of mutations has been reported for CTX patient. So the false positive rate is sort of world record 0 out of 50,000. It's the best performing newborn screen ever reported in the world today. So that's how good the VOS, the Dutch marker is. And this was published in Genetics and Medicine. Okay, so how are we going to get MLD and CTX on the recommended uniform screening panel, the RUSP? So how are we going to get the Secretary Advisory Committee on Heritable Disorders in Newborns and Children to vote to add the diseases? So they have an ASCII requirement. They want to see a prospective pilot study, i.e. with identified newborns. So they made it really hard. It's virtually impossible to do this across the United States with very few exceptions. So yeah, so enter Melissa Wasserstein from Einstein College of Medicine, and she works in a big city called New York City, right? We can put a human live consenter in every delivery bedside of 7 mega hospitals in the New York City area vicinity. 7 humans, 7 consenters at bedside. So you can try to enroll people by social media, advertisement, but it doesn't work very well, as we see in North Carolina, where they're trying to do Facebook type consenting, unsolicited emails. We hear from Don Bailey that participation has been less than 5%. Okay, so to find a CTX baby, we're going to have to screen not for 20,000 babies, but for 200,000 babies, and we don't know the frequency, but maybe it's 1 in 60,000, 1 in 100,000, but we're not going to get there if we screen 10,000 babies. So in New York, Melissa's proven that we can get about 40 to 45,000 babies per year, because Screen Plus this program has been running already for 3 years for other diseases. So Screen Plus is the only functioning prospective pilot program in the United States. Except, I should say, for Massachusetts, they're running a prospective pilot study on Pompe and MPS I. These have already been added to the RUSP. Massachusetts likes to run pilot studies on RUSP-approved conditions just for their own satisfaction in their own state. And they have a kind of a loophole that they can get consent that is not available in other states. I can go into that later if you want. So we think we can get about 200,000 newborns over 5 years. Yeah, so the funding for this is NIH, and then a large amount of money, about half the money comes from a conglomerate of biotechs who have vested interest in these screens. They have treatment or clinical trials. So the diseases that were selected, the criteria are that there be a treatment already or a clinical trial ongoing, because if we find a patient, tell the family it's consented, identified, then we don't want to leave them hanging. We want to give them treatment options. So the subset of New York State blood spots that are in Screen Plus, they're polled in the New York Newborn Screening Lab. We pull them from the whole deck, and we run a megaplex LCMS spec assay that we developed in the New York Newborn Screening Lab on 14 diseases. The first tier screen positives will go in parallel to everybody will be DNA sequenced by the DNA sequence lab in the New York Department of Health. And then for biochemical testing, which will be done in parallel, it'll go to Mayo Clinic, Dieter Matern in Rochester, Minnesota, or my lab at UW. So we're going to do DNA and biomarker second tier. For some diseases like lysosomal acid lipase deficiency, we don't have known biochemical markers. It'll just be DNA. For CTX, we have DNA plus Andrea's marker. For MPSs, we have glycosaminoglycans, for example. So after we're pretty sure the baby has a disease because it's been through 1 or 2, at least biochemical plus DNA or 2 rounds of biochemical plus DNA. We will contact the families and MDs and, get them in touch with treatment centers, clinical trials. And then the second component of Screen Plus is Aaron Goldenberg at Case Western is going to be surveying families for how they feel about this expanded newborn screening. Okay. So the website, so Screen Plus started about a month ago officially. Because of COVID, we lost a year. So here's the website, you can go to it. And here are the diseases. You'll see NLD and CTX on the list. There's 14 diseases. It's a fluid list. There's talk about adding MPS IIIA soon because clinical trials are starting. Canavan disease, some other things we might be adding, amino acid decarboxylase deficiency, molybdenum cofactor deficiency, a whole bunch of other things. The beauty of mass spec, it's the only technology in the world that can handle this kind of biomarker enzyme assay panel in one go. It looks something like this. In 2 minutes, you measure all of these things, including Fred's marker. I just put this slide in. I don't know, Andrea, do we need to wait until we find a new CTX patient in Screen Plus to renominate CTX to the RUS? I'm not sure, to be honest. Here's why. Andrea, if we can provide good input on the other deficits told to us by the RUS folks, like in Israel, when they treat the population at birth, do they do better than the ones treated later? Is the disease spectrum well defined, Austin? But I don't know. Let's say we get to 100,000 in New York. We have 50,000 in Washington, 20,000 in Holland, and we have no false positives, which I'm pretty sure is going to be the case because we don't at 50,000 and we don't at 16,000. And we show that if we take 10 to 12 newborn blood spots from diagnosed CTX patients, if we can get their newborn blood spots and show that our cutoff is we would not have missed any of these, do we really need to find the next one prospectively? And the answer is, as of now, the answer is yes. But remember, Cynthia Powell is the new chair of the RUS committee, and I think she might listen to some of these arguments. To be honest, it's beyond me why we really need to find a new patient. Logically, it doesn't make any sense. At this point, we've proven that the screen works. And why do we have to find the next one just to be an annoying hurdle for no apparent reason? Now, they might argue that you need to show not only that the test works, but that you can put all the medical components in place to get the patient to treatment. So they might say, well, unless you do that, so we think your screen's good, right? We think the false positive rate is low. We think the false negative rate is low, but you haven't proven that you can go through the New York medical system and get this patient treated early. Okay, they might want to see that. Okay, all right, my team and a bunch of collaborators, thank you very much. Okay, done.
That's great. Thanks very much, Mike. And definitely, some points for discussion there for the future and for the committee, the renominating committee. If you want to sign up, please send me an email to join the group. And so for the final speaker today, I'm very happy to welcome Dr. Penelope Bonnen, and we are allowed to run over for another 10 minutes. So thanks to all the speakers for keeping on time, and I'm looking forward to Penelope's talk now. You might be on mute.
Yes. Okay, now do you see my slides? Great. Okay. Moving forward, I'm Penelope Bonnen. I'm a professor of genetics at Baylor College of Medicine, and I'm here today to talk to you about the epidemiology and genetics of CTX. I'll try to go through this as quickly as I can. I just wanted to begin with some basic information that I'm sure many of you know. A rare genetic disease is one that is caused by genetic mutations that are inherited or occur de novo, and they have a very low frequency. Individually, they are rare, but combined, they're not so rare. There are anywhere from 260 to 450 million persons affected globally. And I think it's wonderful to see families and advocates coming together today to learn from and support each other across different diagnoses. So today I'm here to talk to you about CTX. This is a rare recessive disorder. The onset is typically in childhood, and the clinical features are typically thought to be chronic diarrhea, cataracts, tendinous xanthomas, and progressive neurologic dysfunction. It is a rare disease. It has been thought to be an ultra rare disease. And when I started to work on CTX, I was asked the question, how rare is it? And so being a geneticist, I took a population genetics approach to estimating the incidence of CTX. I studied a database of 120,000 exomes from adults who do not have CTX. And so these individuals had the potential to be carriers of pathogenic mutations that can cause CTX when an individual inherits 2 mutations. So we found the population frequency of pathogenic CTX variants, and we also did some bioinformatics to identify variants that were in the database that could possibly cause CTX. And so taking this approach, we estimated the incidence of CTX to have a range of about 1 in 135,000 to 1 in almost 500,000 white individuals, and 1 in 260,000 to about 1 in 500,000 Black individuals. And then we found a consistently higher frequency estimate in Asian populations of about 1 in 100, 1 in 64 to 1 in 75,000 Asians. So this was more common than previously thought. We also did a study on the CTX clinical presentation in families, and we took this project to try to do a systematic evidence review for looking for genotype phenotype correlations. And in taking a family-based approach, we could really, rather than study individual genotypes across unrelated individuals, we looked at the clinical presentation within a family that had multiple affected individuals. We studied all families that had been published in the literature in the last decade. This turned out to be 92 individuals who were comprised in 40 families. And I have a graphic on the right that shows the 12 most frequent clinical features that were observed or were reported in those 92 individuals. And I've listed the top most commonly occurring features here to the left. So the most common clinical feature in those 92 individuals was cataract. And this was followed by cerebellar, pyramidal dysfunction, peripheral neuropathy, and then diarrhea, and then we had an equal frequency of psychiatric features and xanthomas. We also looked at the discordance between family members for these 12 clinical features. So we said these are the top 12 most common clinical features in this cohort of individuals. Now, if we just look in a pairwise fashion within families of affected individuals at these 12 clinical features, how often do 2 members of a family exhibit the same 12 clinical features, either in a plus, they both had it, or a negative, they both didn't have it, that's when they're concordant. But if one individual in the family had it and the other didn't, that would be discordant. And we did this pairwise discordance analysis across all the pairs in these families. And we found that with the most individuals, most pairwise assessments had a discordance of 1. So across all 12 features, they were only discordant in 1 of the 12. And the second most common result was that they were discordant for 2 features. So what we're finding through this analysis is that most patients within a family share the same features. And we did note that patients within the same family may have differing severity in the features that they experience, but our discordance analysis shows that they have most often the same clinical features. So in conclusions, I talked to you today about disease incidence of CTX. It is higher than previously thought. And the clinical presentation within families is more similar than previously thought. There are reports in the literature that say that it's a highly variable disease and that it varies highly even within families, but that's not what our systematic evidence review bore out in this case. Thank you. Thank you for having me today.
Well, thank you so much, Penelope. That is extremely interesting. And as you say, it's kind of not what's been published before in the literature. So we have just 3 or 4 minutes for questions. So please feel free to go ahead if anyone has any questions of the speakers. So I don't see any in the chat, so I will wait for any questions to pop up. I do have a question for Fred. So if everything were to work with your pilot, and you know, you're about 75% of the way through, you know, would there be a timeline? Do you have an estimated timeline for when this might be added to the for the newborn screening for the you know, the national trial?
I honestly, I don't know. Corona has messed up a lot of stuff. So I would say, I mean, I'm already talking with the people that are involved in the newborn screening in the Netherlands. We're closely collaborating. So yeah, I'll try and do my best to get it on the road as soon as possible. But honestly, I don't know. These things take time. These wheels move very slowly, unfortunately. So I would say that, yeah, well, I think 1-2 years or something like that. I hope that we can manage that time frame.
And then I have another question for Penelope. So, you know, this may be a bit further, you know, along than the data that you presented. But, you know, do you think it might be possible then to, you know, have a genotypic variants where you may be able to predict the phenotype? Or is it, so, you know, as I mentioned to Mike yesterday, he was talking about ways, you know, that they would, for MLD, you know, potentially be able to predict the course of the disease from the genetic information. You know, we are, this is one of the challenges in CTX, that, you know, we don't really have that data available. Do you think this is something, you know, that you would be working on in the future?
Sorry, Andrew, were you asking me if I found a genotype-phenotype association? Is that
yes, I mean, if ultimately, you know, this may help, you know, people be able to predict phenotype from the genotypes. So you see there's an association between symptoms in family. So, you know, do you think then that ultimately we'll may be able to take this information and it could help predict a phenotype for specific genotypes?
Well, I think that's certainly worth exploring further, but I think it appears to be evident that we may converge upon and maybe we have already converged upon what the main features of CTX are, regardless of genotype, right? And so I think it kind of depends on the resolution of the phenotype that you're wanting to predict.
Or I think we're more concerned about maybe the age of onset, because the Russ panel, I've heard a few people on that panel say that they're not convinced that CTX is a neonatal disease, and they don't like to approve conditions that are not neonatal. Now, I don't know what neonatal means exactly, and how neonatal it needs to be. A kid that gets sick when they're 5 years old is a pretty bad life outcome, right? And it doesn't, so, but
I think is actually really very important to put greater resolution on. The very first time I went to a conference and talked about CTX, I had doctors come up to me and say, this is an adult onset disease. It's not pediatric onset. So I think you are fighting a bit of history there, right? But I think we do have enough reports in the literature and anecdotal reports to be able to say this is pediatric onset, but I don't know that we have really put together a systematic review that definitively shows that. I think you want to be looking for things like developmental delay. You want to be looking for things like IQ. I think you want to be looking for the neurological component that it's in the literature, but it may not have been sort of presented in the literature in a really concise, systematic way.
I mean, we heard from I think that's the biggest hang up for CTX, is not that we don't have a prospective file, that some people think it's a later onset disease, and it's better suited for a pediatric screen rather than a newborn screen. That's what you're fighting against. But we don't do pediatric screens, right?
Right.
So that was kind of a cheap shot, but yeah. So
I think
the Israeli data is going to be important, that you show that the treatment of these population at birth is helpful.
So Tara has let us know they're going to actually have to shut the session down now. So thanks again so much to all the speakers, and I'm really looking forward to having an in-person meeting next year and interacting. And please, I would, if you're interested in supporting and to generating evidence to help support the resubmission, please get in touch with me, and we'll start having meetings where we could, discuss this further. There's some really great talks today, and thanks very much, everyone. So I think we'll.
Thank you, Andrea.
Shut it down. Thanks very much,
everyone.
Thanks,
Thank you.
Thank you, Andrea.
Thank you so much to our speakers, our sponsors, and our audience. I appreciate it very much for making the virtual family conference this year a possibility. We hope to see you in person next year. Thank you so much.